All right. So the announcements here. So the Unit B module is finished. So there's now a Unit B study guide at the top. All of the lectures for Unit B. The Unit B study guide, if you go into it, you'll find at the bottom there's sort of highlight slides for Unit B, and there's likewise that for Unit A. And those study guides, suggest the things to study. They tell you the course outcomes that are associated with that unit. They have key terms listed. And like I said, I've also taken a small sample of slides from each of the individual slides and packaged them into one spot to make it sort of a one-stop shop for studying. I am, because I was waiting to finish this lecture, I need to still add a couple of questions to this lecture to this comprehension assignment. So I'm just going to make it I'll make it due at the normal due date, but I'll make it available at a week. So I'll finish those questions tonight now that I'm done with this lecture, and then it'll be due Sunday, but it'll be available for no penalty from a week from when I finally post it. And then I'll post an announcement when it's posted. So, but the ARE is still out there, so that's still available. And it's just a reminder to check those study guides, because I think at least as we get closer to the midterm and the final exam, then those will be a lot easier to glance over than going through all of the individual lectures. So that's that. So where we left off, we've been sort of talking about phenotypic variation and where it comes from, genes or the environment. There's obviously a lot of phenotypic variation in organisms that we can see around us. And so by that I mean different color, different length of ears, different curl of the tail, different height, different pitch of the bark, and then behavioral differences too. So a lethargic dog, an aggressive dog, an intelligent dog, and so on. These are sort of behavioral traits, and a lot of variation across those. But the question is, where does that come from? Is it just the genes, or is it the environment that the animal has been brought up in that gives them those differences? And so, that's kind of the question is, to what extent are changes delta phenotype, differences in phenotype, explained by differences in genotype or differences in the environment? And so last time we focused on behavioral tools for teasing that out. We talked about the common garden experiments, where you take two individuals that differ in phenotype, and you're not sure if the reason they differ in phenotype is because their environment's different or their genes are different. So you bring them in to, so you might raise, for example, individuals, offspring, that their local groups, their ecotypes differ, and you might raise them in a common garden, in a shared environment. And if they end up coming out looking the same, then that means, oh, it was the environment that was responsible for that ecotype. If they come out looking different, then that means that, oh, those persistent differences are probably heritable. And the unit of inheritance that we focus most on is the gene. So it's probably genetic. So that's the common garden. So that is a behavioral experiment you can do by moving individuals and then seeing what happens. The other thing you can do is cross-fostering, similar idea. But in that case, you're saying, we notice that there are variations in, say, the call of different birds. So what we're going to do is raise the young of one bird in the nest of another and see if the calls follow the bird, or if the bird in the new home ends up taking on the calls of its new home. And that also allows you to make an inference about genes or the environment. Things that you have to worry about, though, with cross-fostering is that if you do this in the wild, then even though the egg, for example, of one species or one ecotype is with a different species, if they're naturally sympatric, so if they're naturally live in the same environment, the same area, then even though the direct environment is that new nest, which should be different, there is immediately behind that, they can still hear their conspecifics. They can still hear those. So, it may be that the environmental cues that are responsible for the differences in phenotype aren't directly from, mom or dad, but they're from the environment around the nest. And you haven't changed the environment around the nest, you just changed the nest. So that's the reason why cross-fostering often requires a more controlled setup in a lab. Then the other behavioral thing that we talked about were twin studies. The extreme case here, you can have twins, identical twins in the womb, and you can have non-identical twins. So monozygotic and dizygotic twins in the womb and study different outcomes. And if you notice that over a number of different comparisons, a particular trait is more similar among the identical twins, then perhaps it is likely to be genetic. If it's more similar, if the identical twins have the same relationship in that trait as the non-identical twins, the dizygotic twins, then in that case, it's probably the environment that they grew up in, the womb, for example, that they grew up in. And so there are definitely relationships between these different methods. You can kind of see it's all about, taking an organism out of or basically controlling an environment in order to better understand what's left for the genes. That's kind of these behavioral things. So that's where we were. And where we want to go today is to look at more quantitative approaches that allow us to not only maybe decide if something is heritable or not, is coming from the genes or not, but actually figure out where in the genome is it coming from. So before we get to that, are there any questions about the behavioral stuff? Okay. All right, so back to this basic picture, how much of this variation comes from that variation or that variation? That's like the key question here. And so earlier we introduced this term heritability, which was, as a reminder, a proportion of the phenotypic variance explained by the genotypic variance. And so we know that genotypes vary in a population, and we know that phenotypes vary in a population. How do those two variations relate? And it is a property of a trait in a population. So if you are talking about a certain trait or a certain character, you could say like the aggressiveness. Aggressiveness might be heritable in one population. So those that would mean that if you look across that population, you can find some that have a gene and some that don't have a gene. And the ones that have that gene tend to be aggressive. And the ones that don't have that gene tend not to be. And so in that population, that is a highly heritable trait. But the same species and the same trait, if you go to another population, might not be heritable. And you'd say, well, how could that be? Well, it might be that in one population, that trait was under strong selection, and in another population, it wasn't. And so in the population where it's lost heritability, that might mean that it was so beneficial to be aggressive in that environment with that background context that aggression spread, and now every individual is aggressive. And there's the only variation in aggressiveness you get is just due to environmental variation. It's just, one of them, at that point, you're just looking at whoever had a bigger meal in the morning, maybe is more aggressive later in the day. And so in that population, when you're under strong selection, it purges the heritability from the population. And so that same trait in that population isn't heritable. Like the genotypic match doesn't match the phenotypic. But in the other population, where maybe it's not under selection, where it's fine if you're aggressive and it's fine if you're not, then the background genetic variation that actually predicts the phenotypic variation just stays there, because there's no reason for one allele to get favored. And so then you have to wait for genetic drift or some other process. And so that's why you can have two different populations have two different levels of heritability. So it all comes down to a kind of the cartoon of this graph here, where we're going to be expanding on this today, but this is the conceptual model of what this heritability number is. So it's a number between 0 and 1, and it basically represents the slope of this line. And so it's this idea that you have on one axis the variation in the population in the behavior or the trait, but we can say behavior in this class, that you can actually see these differences. Some individuals are aggressive and some are not, some are somewhere in between. There is variation in that. And then if you were to look at their genetics, then you would also notice that some individuals have certain alleles, some individuals have other alleles, and so on and so forth. And so you get a variation in genotype and a variation in phenotype. And each one of these dots represents an individual. So this individual here is very aggressive and they have a particular set of alleles. This individual is equally aggressive, but they have a different set of alleles, and so on. So if you plot all of the individuals from that population, you might get something kind of cigar-shaped like this, and there would be a correlation between the phenotype and the genotype. And that would be an example of where we'd say the genes are sort of beating the environment, and this, in this population, this is a heritable trait. In contrast to that, then you could imagine a different way where you've got the same variation in phenotype, the same variation in genotype, but there's no relationship between those two distributions. So if you're sort of intermediate phenotype, you could be any genotype. And likewise, for any phenotype, there might be a different level of the genotypes you can get, but there's no directional connection between them. And so this would be the case where this variation in phenotype is probably due to the environment, not the genes. So this would be low heritability. So this is sort of a cartoon version of what we're going to do a little more concretely here in just a second. But this is the basic picture I want you to have in your head, is thinking about how much does the character, the trait values vary in a population, and how much do the genes vary in a population, and do those two variations match? So do they correlate? Because you can have variation without them matching. So are there any questions about this basic idea? Yeah. And the genotype can vary. So the genes in the individuals can vary, the alleles in the individuals can vary. And so can then the actual behaviors. And so you might, if you just genotyped everybody, then you might notice that, that somebody is, big A, a bunch of others are little a, some are heterozygous, big A, little a, and you have all these variations all around, and maybe they're equally distributed. So everybody's got that variation. And you might also notice that everybody's got a similar set of variations in whether they sleep 4 hours, 5 hours, or 8 hours a night, or something like that. And you'd say, well, I've got variation in how long you sleep every night, and I've got variation in the genes. Clearly, the genes are determining that. Well, that's the thing. We don't know. We have to actually see, is there a correlation? If you are little a, do you frequently sleep 4 hours a night? If you're big A, do you sleep 8 hours a night? And if you're in between, do you sleep somewhere in between? If that's the case, where the variation in the genes predicts the variation in the phenotype, then we have high heritability. But it might be the case that for every little a individual out there who sleeps 4 hours a night, there's another little a individual out there who sleeps 8 hours a night. So even though there's variation in how long you sleep a night, there's variation in the genes, they're not connected. And so we would say it's not a heritable trait, this how long you sleep at night. Okay. So let's drill down into that a little bit more. And so there are two types of genetic variation that we have to worry about when we do kind of this population genetics stuff. And we don't have to go too far down into this, but I do have to introduce these terms because the easy version of heritability requires us to limit our genetic variation to what we refer to as additive genetic variation. So it makes it a little hard to define what I mean by genetic variation now that I'm going to have these two types, which I'm going to talk about here in a second, but it makes it easier to talk about heritability later. So the two types of genetic variation we have are so-called additive variation, which is very much like what I just told you. So if we're talking about something discrete, like a single locus, a single gene, single locus has two alleles, little a, or little a and big A, and so you can mix them little a, heterozygous, big A, little a, and homozygous, big A, big A, big A. If you've got those three and it's additive, then the number of big A's might determine the level of the trait that you have, and it's just additive. There's no masking effects, there's no dominance effects, there's no epistatic effects, there's no other gene B that changes how A works. Yeah. genetic variation. So variation in phenotype can be due to additive effects or non-additive effects. And so when you have changes in phenotypes that are due to dominance, like you've got a recessive gene and that recessive gene gets masked by a dominant gene, or we have epistatic interactions where without the certain gene, you get a certain expression for one other gene, but then when you add in that other gene, you get a very different type of expression. Those are non-additive effects. So you can't, they require you to understand the interaction between those genes in order to figure out what that phenotype is. So basically, if you were to turn this into a big regression line where you've got the somehow a measure of genotypic difference on the x-axis and phenotypic difference on the y-axis, then there's going to be patterns in those points that come out, and the additive patterns are the ones you can fit with a line, and the non-additive patterns are ones that kind of take other shapes as you swing from across the genotype. And so you can't just, like I was saying, like if it really is an additive, big A, the number of big A's is all you care about, that's great. But if it's actually, well, if you're little a, you get one thing. If you're big A, little a, you get a something totally different. And if you're big A, then you get a third thing that's completely different than that. And it really matters which of those three you are. And there's no relationship between those three. That's like a non-additive effect. And so that isn't captured by what we mean by additive variation. And so when we're talking about breeding, for example, that's usually when people talk about additive genetic effects, because they say sort of like, well, I have a dog that has, this dog has short ears, this dog has big ears, I breed them, and to what extent do I get medium-sized ears? And that relationship between the big ears you bred into that and the medium-sized ears you got out of it, that is what we refer to as an additive effect. Now, there could be non-additive effects hidden in there that are affecting the variation, but for breeders, they don't care about those because they can't really dial into them. So I don't need you to know a whole lot about how you would measure non-additive effects. I just want you to know that in the notions of heritability that we will primarily use, and that most people will primarily use, they're not talking about generically the proportion of all genetic variation, but they're talking about the proportion of additive genetic variation. So, these dominance type effects or whatever are not being included in this heritability proportion that we will primarily use. So that's the big thing. And I know it's a lot of words or whatever, but I have to introduce it just so I can make heritability easier later. All right, so let me dial on here, and then we'll see maybe that's a little bit more what we're talking about here. So the ugliest heritability that we generally don't use, but accounts for all genetic variation, so additive and non-additive, and it's called broad sense heritability, or capital H squared. And so if you had a way to, and you have to use sort of more sophisticated statistical tools to analyze the amount of genetic variation in a population, both the additive kind and the non-additive kind, then you could theoretically measure a trait's broad sense heritability in a population. But what we mainly, and what breeders mainly are going to focus on, is so-called narrow sense heritability, or little h squared. And this is just the additive genetic variation, the proportion of phenotypic variation that is explained by additive genetic effects for this trait. So that's when we talk about heritability, this is generally what we're talking about. But I just have to introduce the difference, because the generic term heritability kind of implies all of the genetic effects. But narrow sense heritability is much easier to measure. And so in practice, when you talk about breeding and things like that, then we're primarily going to focus on little h squared, not this big H squared thing, even though big H squared is kind of truer to the name. So I know it's not going to be that, it's still probably very murky to me what I mean by additive and non-additive. And if you take, it's kind of outside the scope of this course, and that's why I'm not going to focus too much on broad versus narrow in this course. There are other courses that you could take where, evolution courses, for example, where we would actually, sort of be able to plot out, like, let's take a simple single locus gene with two alleles, and let's then actually measure the additive and the non-additive effects at that particular gene to sort of see what the contributions of both the additive and the non-additive effects are to the heritability. And you can do that calculation. That's just out of the scope of this course. For this course, I just need you to know that there is an additive heritability, and it's really, or there's an additive variation, but really the big thing is I need you to know that we're using little h squared. So if you're ever to like look up heritability, it's not the big H we're using, it's the little h. So I know I'm walking around talking a lot about it. I just would say, don't worry too much about this. The reason why narrow sense heritability is so nice is that we can actually estimate it, measure it even, not by having to look at the genes themselves, but just by looking at parent and offspring. Because when you bring two parents together, the variation that you get out in a phenotype that you can study in the parents as well is an additive process. Then this additive variation comes out just by looking at the changes in the offspring phenotype relative to the changes in the parent phenotype. So basically, if you know the average height of parents and for a given offspring, and you have a bunch of different offspring, then you can study the narrow sense heritability of height. So if I were to give that example here, so these are data points where each point is a child or maybe an adult. So these are in 60 some inches or whatever. So each one of these is an offspring, and so each point is the offspring height on the y-axis and the so-called mid-parent height, which is just the average of the two parents' heights on the x-axis. And so this is what I mean by plotting these two against each other. So for every individual in the population, you plot the average phenotypic value, the average trait value for the parents, and you plot the actual value for the offspring, and you get a cloud. And if that cloud, you then put a regression line on it, then the regression coefficient will end up basically the slope of that line, for all intents and purposes, is the narrow sense heritability. So if this trait is not heritable, then this will be flat. If this trait is highly heritable, then it will be a diagonal line. Yeah. The average of the two parents. So if one parent is 6 feet tall and the other parent is 5 foot tall, the mid-parent height would be 5.5 feet. And then, so then if that offspring was also 5.5 feet, then you'd plot a point right at 5.5 and 5.5. And so that's all we're doing there. We're matching the average of the parents to the offsprings. And if you just plot those against each other, and I'm not going to ask you to do this. I'm not going to give you a bunch of data and have you calculate heritabilities. But I do hope that if you were to see a graph like this, you would be able to tell me, does that show high heritability or low heritability? Can you qualitatively, assess the heritability of a trait by looking at data like these? And can you understand how these data were collected? I don't care about the quantities. I'm not going to have you do regression lines. I'm not going to have you calculate regression coefficients or any of that. That's outside of the scope of the class. But I do think in this class, if you were to look at, if I were to give you a graph like this, you'd be able to make sense of it. And just sense of it in the most kind of abstract sense. So is it high? Is it low? Is it intermediate? What would it mean for it to have low heritability? Yeah. That's a great question. So is there such thing as negative heritability? So interestingly enough, so strictly speaking, heritability is a squared value, so it should be positive. However, there are in the end, when people throw these data in, there are tools that calculate heritability. And a lot of times those tools actually have a hard time figuring out what the real heritability is if it's really, really complicated. And sometimes they output negative values. And those negative values have to be interpreted in a certain way that has to do with the signals in your data. So strictly speaking, all heritabilities are positive. That said, if you were to grab a random paper that brought up heritability, it might show you a negative heritability, and but that would indicate that there's something weird going on with the data that prevented the computational tool to actually be able to find a good estimate of heritability. So it doesn't have a physical meaning. Like what you were saying is like, well, what if there's a negative relationship? Like if the parents are tall, then maybe that means that the offspring are short, and that relationship would still be listed as a positive heritability, but the direction of the relationship would be negative. So remember, the heritability is about explaining variance, and variance is always positive. So how much, what fraction of the variance in the offspring can be explained by variance in the parents? That's what the heritability is. The direction of it, like sort of the, I'll say H without the square, that's a slightly different question. And this is just similar to, like, for those of you who've taken a biometry course, if you've learned about regression lines, there's a difference between the regression coefficient, which can be negative, and the R squared value. The R squared value is the fraction of variance explained, but the regression coefficient is sort of the direction of the relationship, and it can be negative or positive. And it's a similar thing going on here. All right, so any other questions about that? This is narrow sense heritability. We can estimate it just by looking in sexual populations, just by looking at parents and offspring, mid-parent values and offspring values, and it's just a line fitting that relationship. Low heritability means that you'll just have a cloud where it'll be a horizontal line. High heritability means you get these slopes. Okay. Heritability is a weird thing. It can differ across subpopulations. Even within a population, there are subpopulations. So this is an example from a paper on houseflies. And in this example here, we have two behaviors, mount and buzz. So these are two sexual behaviors that the male performs when courting the female. And some males mount and some males don't, and some males buzz and some males don't. And so then the question is, does the presence of mount and buzz, are those two things heritable? And so they can look at parent-son heritability and parent-daughter heritability. And so they've gone through and they've done the thing that we just did, where they plotted that line with parent-son, parent-daughter, and then they calculated an H squared value, a heritability value, and then they plotted a confidence interval. So it turns out that when we plot these things, we don't just get a heritability value, we get a 95% confidence interval where the heritability could be lower than this or higher than this, but it's a bound on what the heritability should be. And so that's what they're plotting here. And if you've got a confidence interval that doesn't include 0, then that means that there is a significant heritability. If it does include 0, then that means we can't tell the difference between that heritability and 0. You need to gather more data. So, so what they're showing here is that for parent-son in these behaviors, then you actually get both these behaviors, mount and buzz, are heritable. So they get two confidence intervals on heritability, which do not include 0. They're positive. And so here you have this heritability that the estimate of buzz for parent-daughter excludes 0, so it is heritable. But this estimate for mount, it includes 0 in the estimate for this relationship. So basically, if you gathered more data, it is possible you could find that this is heritable, but it's likely heritable with a much lower heritability. And so this is the case where you've got a strong signal of heritability in parent-son, but you've got a weak signal of heritability in parent-daughter. So even among subpopulations that live with each other, then you can have heritability that differs. Heritability that differs across sexes. So it's kind of interesting there. And you have to think, well, how does that happen? And I just remember, selection does tricky things to heritability. So, if something is important, if it is advantageous or disadvantageous, if it really hurts you to have it or really helps you to have it, then that trait will probably be selected for. And the more selective pressure that's on that trait, or against, but either way, the more selective pressure on a trait, the less heritable it will become. Well, a trait that is good for one sex may not have any value for the other sex. So the one that doesn't have the value may have high levels of heritability still, because there's no selective pressure acting on that, taking advantage of that heritability. But the one where it is good for, then the selective pressure is taking advantage of the heritability, and it's gradually reducing the heritability. So because different traits have values that can vary by sex and maybe by other things, then heritability can vary by sex. Any questions about that? So that might be actually, yeah. All right. So, with that, let's do one of these. If I can find my mouse. There it is. I'm just starting. I'll let you know. Okay, 331. All right, I'll start the multiple choice now. All right, so question is, what type of genetic variation does broad sense heritability include and narrow sense heritability exclude? So there's a type of genetic variation that could explain phenotypic variation. Broad sense heritability includes that type of genetic variation. Narrow sense gets rid of it for simplicity. And so the answer I'm looking for is either phenotypic, additive, non-additive, or summative. All right. So it looks like we're settling down here. I'll go ahead and stop this poll in, say, 15 seconds. 5 seconds. All right, I'm going to stop the poll now. So look at the results here. Looks like we've got 7% for A, 10% for B, 76% for C, and 7% for D. And I was looking for non-additive. So that's the thing that this broad sense, most generic heritability has these weird non-additive things. It tries to account for them as well, dominance, epistasis, and so on. But the generic heritability, where you forget that genes can interact in these weird masking ways and have these interaction effects, those kind of simpler things, is this additive, and that's the narrow sense heritability. So those are the two types of genetic variation, additive and non-additive, and that's it. And so broad sense includes them both, and narrow sense is only additive. Okay. All right, so going back to what I was saying before, the maybe. Okay. All right, so I already mentioned this, but would you expect to see to frequently find a highly heritable trait that also provides a strong selective advantage or disadvantage? Yeah. Yep. Okay, well, that's exactly related to this. So does anybody else have an answer to that? Why does selective pressure reduce heritability? When we have a guess at an answer for that? Yeah. Right. So there's an example there about clause and no clause. And so we could say there's a genetic basis for having clause. And if there's a genetic basis for having clause, then those who will have clause in an environment that favors clause will pass on the genes for clause, and those that pass on the genes for no clause won't have very many offspring. So now over time, you still will have individuals that have the gene for clause that maybe they can't find enough of the nutrients to produce the cuticle, or they just ground down all their claws. There's all these environmental reasons why they might not have claws. But at that point, that's just environmental variation. So the reason why selection, good or bad, purges heritability from a population is because those that because selection just generally reduces genetic variation. So as you take a population that has a lot of variation in the genes and a lot of variation in the phenotype, then you potentially have the potential for high heritability. But as selection marches on, it's going to get rid of the genetic variation. You may still have variation of the phenotype just because there's G by E, the environment is still there. But the variation that you're left with in the phenotype is going to be due not to the genotype, because anyone with a gene for an advantageous trait, that gene will have a greater reproductive advantage and will flush out the other genes so that you have less genetic variation. So that's kind of what I'm getting at here, is that you wouldn't frequently expect to find a highly heritable trait. So if you calculate the heritability of that trait, it would not have a high heritability score, a high H squared, if it also was under strong selective pressure, if it had a strong benefit or cost. And that's just because, at equilibrium, like maybe this happens periodically, but shortly thereafter, then gradually if it has an advantage, it's going to grow in representation. If it's a disadvantage, it's going to fall in representation. So at equilibrium, then any of these traits that any of these genetically defined traits that have a high selective pressure are going to be sort of operated on, and eventually that genetic variation is just going to get purged, because that's what natural selection does, is it reduces genetic variation. So is that clear? Are there questions about that idea? I think I summarized that here. So seeing traits often, highly heritable traits often, indicates that the trait is unlikely to be under much selective pressure. So that's kind of that's what, being an animal behavior person, that's kind of the interesting thing, is that if you can look at a population and you can say, I notice that some of these animals have really loud calls and some of these animals have really quiet calls. Some do mating this way, some do mating that way, some bury things here, some things go up the tree. If you notice there's a bunch of variation, and then you can say, I'm going to see how heritable this variation is. So there's a guy, Blaine Cole, who studies seed harvester ants. And he noticed that you can take seed harvester ants and you can give them tasks. And you can give them like little bits of paper, and they'll sort the paper into piles. And then you can give them little marbles, and they'll sort the marbles into piles, and so on and so forth. And he found, interestingly enough, that although there were ant colonies that were good at sorting paper and good at sorting marbles, and also ant colonies that were bad at sorting paper and bad at sorting marbles, sorting paper was not heritable, but sorting marbles was. And so because he could watch these ant colonies over their generations and then give the offspring the same challenges. And then that basically was saying that if a parent was good at sorting marbles, then it was very likely that the colony that got a daughter colony would also be good at sorting marbles, and so on and so forth. But sorting paper wasn't the case. And so that's kind of interesting, I guess, but what that tells me about sorting marbles is it was highly heritable, but there was still that variation in the population, is that apparently it doesn't matter if you can sort marbles or not. Because, the ant colonies that could sort marbles were doing just as well as the ant colonies that didn't. So even though I see that variation, it's interesting to know, where did it come from initially? Why is this variation? But it could be phenotypic inertia. Those are those other things we talked about, a little while ago. They're kind of stuck in that population, and and they just didn't go away because now there's just no selective advantage for that. So it's a highly heritable trait, but selection's not acting on it, and that's maintaining the variation. Now it's interesting that sorting paper is not highly heritable. Now, what he didn't study, but he might have been able to study, is the ant colonies that are good at sorting paper, are they also happen to be good at, say, finding seeds? Maybe for whatever reason, the task of sorting paper is very similar to the task of finding and securing seeds. We know that seed harvesters need to secure seeds, so maybe the lack in heritability in sorting paper actually reflects a lack in heritability of finding seeds, because every seed harvester needs to find seeds. So the seed harvesters that couldn't find seeds, the heritable part of that got flushed out of the population, and all that you're left with now are seed harvesters that have a genetic predisposition to finding seeds, some that happen to be a little better at it, some that not so great at it. But any variation in finding seeds is environmental variation, not actually lineage variation. So that's kind of an example. Is like, as a behavior person, I'm really interested in how heritable these traits are, because they give me an insight into the ultimate causation. For example, why is this trait sticking around? Is there a benefit to this trait? If there was a benefit, how the heck is it so heritable? Highly heritable traits, if they have a benefit, if you're at equilibrium, probably that doesn't make a lot of sense. So if you're at equilibrium, they're highly heritable, maybe they don't have a benefit. That's something that maybe you can take from that. So there are questions about that basic idea that that we can use heritability to give us an insight into whether we think that trait is under selection. Yeah. That's what we're about to get to. But yeah, so what I mean by out of equilibrium, and I kind of didn't really define that here, but is this idea that if something happens, so there's a flood of genes that flow in from a nearby environment, introducing a new trait that no one's ever seen before, there's going to be a period of time where that trait dukes it out with the existing trait values in the population, and eventually comes to some new equilibrium. Maybe the new trait will sweep the population and take over. Maybe they'll coexist or whatever. But initially, there's going to be a response to that that's a transient response. And so what I mean by out of equilibrium is that the allele frequencies aren't changing that much. That's what I mean by equilibrium. Out of equilibrium means that if you come back generation to generation to generation, the allele frequencies are going to be changing significantly. So if you're out of equilibrium, that means evolution is happening. Evolution is just a change in allele frequencies. If you're in equilibrium, then that means that the allele frequencies are staying stable over time. And so that gets to the question of, how do we determine if we see this, how do we know it's one or the other? So how do we basically know, how close are we to equilibrium? If we're close to equilibrium, maybe we can start making inferences about selective pressure. If we're far from equilibrium, then maybe this is a transient case. And if we come back a little bit later, then the heritability values are going to change. And so that's what we want to get to next here, which is Hardy-Weinberg. So how many of you have heard of Hardy-Weinberg before? Okay, so you've probably, those who've taken genetics, maybe have to a bunch of this sort of, so it's going to be kind of a review, but the idea here is that you can go out and you can actually measure allele frequencies in a population. And if it's a simple single locus gene with two alleles, then you can measure the frequency of one allele as P, and the frequency of the other allele is Q. It's just 1 minus P. And Hardy-Weinberg says that if you're at equilibrium, so if there's no evolutionary pressures acting, so no natural selection, no migration, no drift, none of these things that could change allele frequencies, then what is an example proportion of genotypes that you would expect to see in a population that would just sit there stably? And that, as again, this is probably review, in this particular case, you would expect to see whatever that proportion of the big A allele is, the AA genotype would be P squared, the little a genotype would happen Q squared, and the heterozygous genotype would be 2PQ, and all three of those would add to 1. And so if you go out and you sample a population, you can very quickly tell, for example, the heterozygous, is the amount of the distribution, the frequency of this heterozygous genotype in the population, does it equal 2PQ? If it does, then, or if it's very close to it, then I can probably say that this population is not changing its allele frequencies. It's probably at equilibrium, and high heritability values probably are not under much pressure, or else we wouldn't be at equilibrium. But instead, if I find that if I sample my population and I'm not at Hardy-Weinberg, and nobody's ever at Hardy-Weinberg, but Hardy-Weinberg is like a baseline. And so the closer you are to that baseline, then sort of the closer you are to equilibrium. And if you are close to Hardy-Weinberg, then that, or if you aren't close to Hardy-Weinberg, then things are changing. If you are close to it, then you're sort of suggesting the diversity that you see in that population is likely to be maintained. And this is an example here that was done with this. I think this is a bulbul. So this cute little bird here, this paper that was referred to in the text, they do exactly that. They go out, they sample a bunch of birds, they genotype these birds, they do these high throughput sequencing, and they see how close is this population to Hardy-Weinberg, and they check some other things too. And the reason they do that is because there are some conservation challenges that make it very important to know, is the diversity that they see in this population, is it stable, or is it actually actively changing? And they find that the distance this population is away from Hardy-Weinberg is very similar to other birds. So they say that the diversity that is currently in that population seems to be being maintained. So that's an example of how you can use Hardy-Weinberg for this kind of useful thing here. But generally speaking, no populations are Hardy-Weinberg, but it's kind of like, a baseline. All right, questions about that, about Hardy-Weinberg, or about this idea of equilibrium? I'm not going to ask you to do 2PQ or anything, but I want you to know that Hardy-Weinberg exists, and I want you to know that theoretically it's possible to measure populations' distance from Hardy-Weinberg. How much are they in agreement with Hardy-Weinberg? And that tells you, can help you figure out the selective background in that environment. Okay, great. All right, so there's one last thing that I want us to then talk about where we're done with genetics, behavioral genetics, and go on to learning in the next unit. And so we've been talking about heritability, but we haven't been directly talking about the genes associated with those behaviors. So how do we start identifying the genes that are responsible for complex behaviors? And that gets back to this idea of quantitative traits that we talked about earlier in the unit. So remember, you can have traits like, discrete traits that, is my earlobe connected or not? Do I bark or not? But many traits, especially behavioral traits, are quantitative traits, which means the values you see in the population vary over a continuous range. And due to reasons having to do with the central limit theorem, then typically the values of these traits in the population happen to be on bell curves. So it just happens to be that the height of, people's height tends to be on a bell curve. Other quantitative traits, height, weight, behavior like shyness, degree of shyness. So there's a whole way to assess whether an animal is bold or shyness. It's in this realm of behavioral syndromes and personalities. And these are all values which come from continuous range. And if you look across a population, then all of the individuals fill out kind of a bell curve like this. And the variation that you see in this bell curve comes in part from environmental effects, because those are also going to naturally vary. But they also come from a combination of different genes. So I mentioned this before, when quantitative traits like Darwin was really into smooth variation. That's like natural selection is all talking about heritable variation. And with Darwin, it was like the trait changes a little bit. It's a very slow process. And yet Mendel was all about these traits that have just two discrete values. And so Mendelian inheritance was the thing that sort of made the most sense over time, but people had a hard time until quantitative genetics trying to understand how you go from Mendel's inheritance to Darwin's variation. And this is basically it. It's the idea that, well, there's not just one gene, there's multiple genes, and those genes then they can express in different amounts collectively together, and a complex behavior isn't going to be determined by one gene. It's going to be determined by a symphony of genes that all do different things and participate in other things as well. So there's not one gene to one trait. There's, this gene participates in these 50 different traits, but it happens to participate more in trait A than trait B, and so on and so forth. And so the mapping from genes to phenotype is much more complicated, and that's what quantitative genetics tries to wrap its head around. And so that's what we want to know, is how do we figure out which genes are participating the most in which behavior? And so this is what we would like. So there's this notion of quantitative trait loci, or loci, the plural of locus. So what we would like is to say, let's lay out the genome on the x-axis here. And so this is the left of the genome, and this is the right of the genome over here. And every spot on this x-axis is a position. So you can think of it as a gene. And ideally, what we'd like to do is to have some way to sweep across this entire genome and ask, how likely is this behavior to have some aspect of this gene contributing to it? And if you were to get this sort of green line that would sort of suggest you get these two bumps here, then you could say, well, then this position and this position, those are the two quantitative trait loci that are contributing the most to this behavior. And most behaviors, if you do this analysis, and we'll talk about what this analysis is in just a second, have 3 or 4 of these bumps per behavior. And they have different levels. So this, for this particular trait or this particular behavior, this position on the genome is much more likely to be involved in the behavior than this one. But both are needed to get this behavior working. So this is the notion of the quantitative trait loci, or the QTLs. And what we want to do is to try to figure out where these QTLs are, to do so-called QTL mapping. So before I go and show how we do QTL mapping, are there questions about this general idea of what I'm talking about here? So the idea here is you sweep across all positions in the genome, all genes, and then you get this green line, and the 2 peaks of this green line correspond to the 2 red dots here. So these are the 2 positions on the genome that are most likely to be contributing together to that behavior. This is the odds that this QTL is contributing to the behavior that we're studying. So it's the odds that this portion of the genome is contributing to this phenotype. All right, so let's see how we get one of these graphs. So the basic idea here is the first thing you have to do in QTL mapping is you take a genome of an animal and you then have to establish a set of genetic markers. These are landmarks that have nothing to do with your behavior at this point. They're just kind of like mile markers on the road that is the genome. And so a lot of times these nowadays are these so-called single nucleotide polymorphisms. So there are, along your genome, you can find a particular spot where if you're to look across individuals, then my nucleotide, A, might be different than yours. So we're going to use T, like there would be a difference in this very, very tiny location. Now, they don't have to be SNPs. They could be, and that's how we pronounce that, SNP, SNP. But there are other types of markers. But the idea is you find little sequences that tend to vary across the population if you're lucky, but that you can identify them. And then so you can sort of say, well, this sequence is, these sequences fill out this whole, in this case, this is the whole genome, or in this case, this is chromosome 5 of this particular animal. This is, I took it out of a paper about potatoes. So this is the 5th chromosome of a potato, and these are the 50 markers that they put in that chromosome 5 for that potato. But you do it with any organism or whatever. Again, these markers, they're just landmarks that through molecular methods, you have found these markers that are spaced across the whole genome. Now, the important thing when you choose these markers is that ideally, so what we've got here is this is our chromosome 5 generically for this potato. These are 4 different homologous chromosomes. And so if I were to go across different potatoes and I were to grab the chromosome 5, or one of the 2 chromosome 5s, one of the 2 sides of chromosome 5 on that particular potato, then what we can see here is across the different potatoes here, we get different expression of these different markers. And so we see that not all of these 50 markers show up in all of these potatoes. And that's what I mean by they vary across the population. So, some potatoes have some of the markers, some potatoes have others. And so, like, I can see here that this says 86 right here. So at location 86 in chromosome 5, I've got this marker called SSR5-38-2. Well, if I find 86 in this general map here, it also says SSR5-38-2. And so, so that's each one of these is showing up here, but not all of them are showing up over here. So that's ideally, I just want to choose markers that will vary across individuals. That's one of the first things I need to do here. The presence and absence of these markers in my individuals is what's going to help me map the QTLs to my behaviors, which I'm about to get to. Yeah. You can think of these as 4 different individuals. You can think of this as so if you think about, you've got pairs of chromosomes. Well, in this case, they're potatoes, so it's tetraploid. But if they were you and me, then you've got 23 chromosomes, 22 of them autosomal chromosomes. Each one of them is a pair, and so we're looking at one of that pair of chromosomes. This is mine. This is yours. This is Anushka's. This is Kendra's, or something like that. Mm-hmm. Yes, that's right. So these are all of the markers I can look for. So those of you who've ever done a PCR or whatever in a lab or something, that's in the old days, you just had basically a bunch of these that then you would then say, which of these are present in my sample? And you can do that by amplifying the expression of all of these and these things. And if they exist there, then you can say, that marker is in this individual. That marker is not in this individual, and so on and so forth. So these are all my markers that are possible. These are the markers as they show up across individuals. So I choose these markers, these landmarks. Okay. And then what do I do with these? Well, yeah, so ideally, like I was saying, that these are, this is a marker I chose, and in one parent, it takes one value, and this parent, it takes another. So ideally, that if these markers are a single gene, then there are multiple alleles of that gene. And that's kind of also what I'm trying to say here, but in a different way. So it doesn't always happen. So this marker happens to have across these two parents the same allele, but hopefully you've chosen enough markers that if you were to pick any two parents, a lot of the markers are going to differ. That's the hope when we choose these markers. And the trick here that we're going to see in a second is that our QTLs, are plausible places where our behavior that we're chasing down is likely to be located, is going to be close to some of these markers. So close that sexual recombination won't be able to break them apart. And so if you can find the markers and correlate the markers to the behaviors, you'll be able to infer the QTL's got to be close by. So that's what we're going to see here in just a second. So then step two, now that I've had all my markers, I get two parents that differ in phenotype, blue phenotype, orange phenotype, and I cross them and I get a bunch of offspring out that vary in the phenotype and because it's a quantitative trait in a wide variety of ways. So, short, tall, intermediate, whatever. So I get a bunch of phenotypic variation out of the offspring. Now, remember, I also am going to have genotypic variation in the offspring as well. And so in step three, I now need to then correlate the genotypic variation in my markers with the phenotypic variation in this cross that I did. And so that's where I get something that looks like this. And so now there are programs that do this for you. So I've got you feed into the program all of your markers, and the markers are they know where the markers are on the chromosome, and they can see which markers show up in which offspring, and they can see how much of the quantitative trait shows up in each offspring. And so using that, they can calculate what's known as an odds score, a log odds score. Doesn't matter, just think of it as an odds, and so the higher the number, the more likely it is, the lower the number, the less likely it is. And so on the x-axis here, it's the position across the chromosome. The red is the odds score. So it's saying based on whenever I see the trait and whenever I see the markers, which I know are at a particular spot in the chromosome, then this trait shows up frequently, infrequently, and so on. And so it can use those landmarks to create odds of what is the likelihood that the quantitative trait that is determining that behavior is nearby those landmarks. And that's what creates this map here. And anything above sort of a significance threshold, then you'd say these are the QTLs for that trait, those peaks. That's how we get QTLs out of QTL mapping. Okay, are there questions about that? So I think I have a little summary of this here. So, the key elements here is that I have a small but complete set of markers. I have the ability to do breeding experiments to create phenotypic variation, and I can then correlate the phenotypic and genetic variation to identify the likely locations of the QTLs. That's QTL mapping, and it helps me figure out for a behavior where roughly on the genome is that behavior coming from. So questions about that? Okay. Now, we can do QTL mapping in honeybees really easily, all sorts of animals really easily. But it's not so easy to do for humans. Why would we say that? Like, why is it probably hard to do QTL mapping in humans? Pardon me? The ethical side, yeah, we can't do a lot of crosses in humans, right? We can hope that that we can analyze markers and crosses, and we hope there's a natural experiment that's out there, but we're not going to force people to breed in order to do this. So, but we would like to have something similar, and that is where I want to finish off here, is introduce one more concept in gene mapping called GWAS, or GWA study. So GWAS is Genome-Wide Association Study, and this has been facilitated by the computational power that we now have. This isn't that new, but it was something that certainly it came up, I don't know, 10, 15, 20 years ago, became very, very popular, and is now just sort of very easy to do, well, relatively easy to do if you have the data. But the basic idea here is we can get rid of the breeding. So it's a similar idea as QTL, but there's no breeding involved. And so what we're going to do is we're going to choose a gigantic number of markers, orders of magnitude more markers than you have in QTL. So in QTL, you might have 1,000 markers. With GWAS, maybe it's 10,000 or 1,000,000. We're just going to choose a bunch of markers, usually SNPs, single nucleotide polymorphisms, that vary across the population. And then we're going to gather natural genotype and phenotype data from a huge number of individuals. So like in humans, a huge number of people. And usually when you do human studies, it's phenotype first. And so it's kind of like, well, we'd like to figure out what's the genetic basis of ALS, for example. And so we're gonna choose populations where we know we're going to have a lot of representation of ALS and a lot of representation of those who don't have ALS, as an example. And we're going to get a lot of people, and we're going to genotype or at least get the marker presence and absence data for all of those people. And with all of that, then we can just use computational power to basically say how likely, if we discover a marker in someone with the trait, let's say ALS, how likely is it that that marker is there by chance? And that's going to give us a p-value for every one of those positions. And so, it's kind of like a t-test, a p-value. So if it's less than 0.05, then that means that it is very unlikely that that marker would be there just by chance based on the background of the population there. And then that gives us something that looks like this, which is going to allow us to say which one, the high peaks here are likely to be involved in the trait. The low peaks here are just the background. Yeah. It's just good old-fashioned stats here. I mean, you certainly can do a bunch of data mining sort of methods, but the methods here are nothing special. Like it's not like like the models that are used to do GWAS are relatively straightforward to write down. It just takes because there's so much data, it just takes a long time to run them. It's like running an Excel spreadsheet that has 50 million lines. If you make one change and wait for all the other cells to trickle all that through, it may take a day for the spreadsheet to settle down. That's what I mean by computational power. Yeah. Mm-hmm. Well, so the high parts here, these similar to QTL, these positions, so each one, what this is sort of saying here is that these markers that have these high, this is a logarithmic transform of a p-value, but it's sort of the same idea here, is that the higher this number, the less likely that location is there in the trait just by chance alone. So if we look at individuals with the trait, with ALS, without ALS, then they're going to find that some of them have the markers and some of them don't. And so the question is, then you could say, well, who has the markers, or what markers are found more in people with ALS than not? And so for each marker, you basically can calculate a p-value, which represents, is your presence here, what is the probability, what is sort of the likelihood that, I don't want to use the term likelihood, but this p-value represents, the probability that you're just there by chance as opposed to being there because of the trait. And so when these numbers are high, then that gives us spots on the genome to look for candidates that may be involved with this, just like they did for QTL. But we didn't have to do breeding to generate the variation because we just had a natural population with a lot of variation in it already. So that's the way GWAS is. So I can put these things together and say, to compare them, QTL, you don't need as many markers, you don't need as much computational power, but you need breeding to generate the variation. With GWAS, you just take a background population, so you don't have to, it's observational. You don't have to do an experiment. but you need a lot of observational data and a lot of data per individual and a lot of computational power to generate all of these numbers. And those are the two basic ways that I want you to know of gene mapping. You don't have to know all the details. The biggest thing to know is small number of markers in breeding, large number of markers, and no breeding. That's the biggest difference, but otherwise they're doing roughly the same thing, trying to find which genes are contributing collectively to a trait. All right, well, I don't want to keep you too much longer. I know there's a big debate going on. So if you have questions, feel free to come up after, but let me start this one last multiple choice here. And then feel free to head out of here. So which of the following is a gene mapping method that uses breeding experiments to create offspring with phenotypic variations? I think I missed a word there, but which is the breeding genotypic gene mapping technology? A, B, C, or D, QPL mapping, GWAS, cross-fostering, or narrow-sense heritability? That's all I've got for you. So we'll see you on Thursday for the new unit.